So our next speaker is Aniek Janssen, and she'll be talking about deciphering double-strand break repair in heterochromatin and euchromatin using an in vivo Drosophila model.
Hi everyone, I'm Aniek Janssen. I'm a postdoc in the lab of Gary Karpen. First of all, I would like to thank the organizers for giving me the opportunity to present our work. So if you look at a nucleus, you don't have like one homogeneous mass. There's a lot of different chromatin regions within the nucleus, and two very distinct chromatin domains are heterochromatin and euchromatin. So heterochromatin, as Sally Elgin was already explaining, has a lot of repetitive sequences, is highly compacted, and you can mainly find it at telomeres and pericentromeric regions. And it also contains a very high enrichment of H3K9 methylation, as well as heterochromatin protein 1 or HB1A. In contrast, euchromatin contains a lot of active genes, has a more open conformation, and in general a little lower H3K9 methylation as well as HB1A. So cells are continuously being exposed to to DNA damage, such as irradiation or endogenous defects, such as replication defects, that can lead to double-stranded breaks. And the two main repair pathways for double-stranded breaks are homologous recombination, in which a template is being used to very precisely repair the DNA damage, or non-homologous end joining, in which the two ends are just ligated together, and this could result in small insertions and deletions at the break site. So homologous recombination is active at the S and G2 phases of the cell cycle, whereas non-homologous end joining is active throughout the cell cycle. So a main question in our lab is, how does a cell actually deal with double-stranded breaks within these different chromatin environments? So a lot of the work, or a lot of things that we know from double-strand break repair in heterochromatin comes from previous work from a previous postdoc in our lab, Irene Chiolo, who is now a PI at USC. And she took Drosophila as two cells, irradiated them with 5-gray, and obviously she could see DNA damage foci coming up everywhere, which resemble double-stranded breaks. And then specifically the DNA damage foci within heterochromatin would move to the periphery of heterochromatin and get repaired there. So the main repair pathway that is thought to be active in heterochromatin is homologous recombination, has also been shown in mammalian cells. But this could, of course, if you think about it, be potentially very dangerous because there's a lot of homologous repetitive sequences nearby. And if you have misrepair by homologous recombination, this could result in acentric or dicentric chromosomes. So the hypothesis that we have is that this peripheral movement actually prevents aberrant recombination between repeats. But we still have a few questions left, and the first of that is, can you recapitulate all these findings, so the HR dependency and also the specific movements in an actual organism? And is this something that we would only see with irradiation, or can you also see it with a single break? And on top of that, it's very important to actually do sequence analysis of repair products to really determine what the repair pathway is that's being used in heterochromatin. And that has not been done for heterochromatin so far. So yeah, we wanted to create an inducible in vivo system to, first of all, can we see this movement of the double-stranded breaks as well, single double-strand breaks? What are the contributions of each double-strand break repair pathway? And then if HR is being used, what are the templates that are being used? Are it the homologs or are it the sister chromatids? So we collaborated with Jan Larock on this, who is a PI at Georgetown University, and she already developed a really nice system called DR-White in Drosophila. And this is what the construct looks like. So you have an upstream Y gene, which encodes for the red eye color in flies, but this contains an IC cut site right in the middle of the gene, which creates a premature stop codon, so you cannot have an active transcript. Downstream of that, we have a DS-Rep marker, and then downstream of that, we have an IY gene, which has a 5' and 3' deletion, so again, cannot lead to an active Y transcript, but it does have the wild type sequence where the upstream Y gene has the IC cut site. So if you express IC, you can induce a single double-strand break in the upstream Y gene. And we can then, by sequencing this upstream Y gene, we can determine the use of different repair pathways. So for example, we can look at homologous recombination. So if you have a single double-strand break in the upstream Y gene and you could repair with the downstream IY gene by homologous recombination, you end up with a wild-type Y gene, which we can identify by sequencing. And the other possibility is we can identify non-homologous end joining products, because imperfect non-homologous end joining products in which you have small insertions and deletions, and we will also be able to identify those by sequencing the upstream Y gene. So we integrated this DR-White in 3 euchromatic insertion sites and 6 heterochromatic insertion sites. So for euchromatin, we have 2 insertions in chromosome 2 and 1 in chromosome 3. And for the heterochromatic sites, we have 2 in chromosome 2, 3 in chromosome 3, and 1 in the X chromosome. So on top of these different insertion sites, we also wanted to have an inducible IC expression. So for that, we coupled IC to a degradation domain, which has been developed by the Wanderless lab at Stanford. So you get continuous degradation of your IC protein, but then when you add a stabilizing ligand, in this case trimethoprim, you stabilize the degradation domain and you get expression of IC, which you can see in the pictures below if we treat an eye disc with trimethoprim. So the first thing we wanted to do is live analysis. Do we see movement of single breaks to the periphery? So for that, we again used the DR white, different DR white insertion, expression of ECDHFR IC by putting the discs into trimethoprim containing media. And then we have 2 fluorescent markers, one HP1A, heterochromatin protein 1A, tagged to RFP, so to visualize the heterochromatin domain. And then to visualize DNA damage, we tagged mu2, which is an MDC1 homolog. We tagged it with YFP. And then we just categorize the different foci. What does their movement look like? So one category is in which we see, so the red is the heterochromatin domain, we see the DNA damage focus coming up outside of the heterochromatin domain and then resolving. And then indeed, sometimes we observe DNA damage foci coming up in the inside of the heterochromatin and then within 20 minutes move to the periphery of the heterochromatin domain. And a category that we also observed a lot was DNA damage foci coming up at the periphery, just staying there and get resolved. So if we quantify this in a few euchromatic insertion sites of the DR white integration, the majority comes up on the outside, 82% is what we expected. If we then look at heterochromatic insertion sites, there's much more of a spread, but we definitely see DNA damage foci coming up in the inside and moving to the periphery, and also a big chunk of them come up at the periphery and get resolved there. So we can indeed recapitulate movement to the heterochromatin periphery with single breaks in vivo. And then the next thing I wanted to do is do actually double-strand break repair product analysis, so sequence analysis. So for that we take 30 instar larvae that contain DR white, that express IC, we feed them trimethoprim so you get expression of IC, and then we just PCR the upstream white gene in a DR white construct and sequence it with Illumina sequencing. And then you can look at this graph. So on the y-axis is the total percentage of identified repair events. In black is HR products, so homologous recombination, and in gray is non-homologous end joining repair products. And then you can see for the 2 euchromatic and for the 3 heterochromatic insertion sites that we looked at here, that we see actually quite a lot of non-homologous end joining products. So around 70% of all the products we see are non-homologous end joining products, and 30% are homologous recombination repair products in both U and in heterochromatin. So to confirm this role for both heterochromatin, HR and non-homologous end joining, we wanted to inhibit HR by knocking down RAD51 or inhibiting non-homologous end joining by knocking down Q70. And the analysis that we did is we did live analysis. Can we see a delay in the repair of double-stranded breaks by looking at mu2 foci? So again, we use our live system with the 2 fluorescent proteins. And then here you can look at a euchromatic insertion site. On the y-axis is the percentage of cells with 1 mu2 focus. So we start off with 100%. And then the black line is the control situation. And on the x-axis, you can see the time in minutes from the mu2 focus appearance. So yeah, you can see that around 50% of the DNA damage foci disappear in around 110 minutes after their appearance. But then if you knock down RAD51, which is the gray line, you can see that there's a clear delay in repair of these double-stranded breaks within this euchromatic insertion site. And then we can see the same trend in a heterochromatic insertion site when we get rid of RAD51. And then if we get rid of non-homologous end joining in a euchromatic site, again, we can see a shift. So the gray line is the Q70 RNAi. You can see a shift in a delay in the repair. And that's the same trend that we see if we knock down Q70 in a heterochromatic insertion site. So indeed, recapitulating the role for non-homologous end joining. So quick intermezzo, the single breaks, yes, we do see them moving to the heterochromatin periphery. We see that both HR and non-homologous end joining are involved in U and heterochromatin break repair. And then the final question I would like to talk about today is what templates are being used to actually be able to get safe homologous recombination repair in heterochromatin. So we know that homologs in Drosophila are very closely associated throughout the cell cycle. And indeed, the lab of Kent Golik already showed a long time ago that homologs can be used in the repair of double-strand breaks within the male germline. So to look at whether the homologs are actually paired when we induce a double-strand break, we developed a different system. On one homolog, we introduced a set of leg-O repeats next to an IC cut site, and on the other homolog, we introduced a set of tet-O repeats. And then if we do fish in the absence of any breaks, we obviously, in blue it's the tet-O, and in red it's the leg-O. You can see that they are very close together, the two homologs. And then if we do induce a break, they're still very close together. So yeah, we did not see any significant differences in the leg-O, tet-O distance with or without a double-strand break in either euchromatin or in heterochromatin, indeed indicating that the homolog is present and could be used in U and in heterochromatin. So for that, we introduced the last system that I'm going to talk about today. So we actually wanted to quantify this, the use of the homolog. So for that, we have the DR-wide system on one homolog, the one that I explained before. And on the other homolog, we only have the IY gene, so the downstream repair template. And then we did put 2 single nucleotide polymorphisms around the repair site. So in the case of repair with the homolog, we will be able to see the SNPs coming up. So that's what we quantified here. So on the y-axis, the percentage of HR products with the SNPs, so the percentage of HR product that use the homolog in 2 euchromatic and 2 heterochromatic insertion sites. And it's around 3 to 8%. It's not a massive amount. So this indicates that the homologs can be used, but the majority most likely uses either INCIS or the sister chromatid to repair their double-stranded breaks. So yeah, to end with a model on heterochromatin double-strand break repair and also our future directions. So if you get a break in euchromatin, we can see in our system both HR, non-homologs, and joining being important for this repair. And then if you get a break in heterochromatin, we see the DNA damage foci moving to the periphery. And there they can do either HR with the sister, INCIS, or with the homolog, and thereby do safer combination. Or non-homologs and joining actually in a lot of the cases, which is more imprecise, of course, than homologous recombination, but you will be evading the problems that you have with all the homologous repetitive sequences around. And then, of course, we are interested in what are the roles for alternative pathways, and we haven't addressed them just yet. And then we also would like to do more higher resolution imaging and see what the role for chromatin proteins and modifiers is in all this. And then, yeah, the final question that I want to mention is, of course, why do we see less HR and more non-homologous end joining in the flies when compared to irradiation of cells in culture? So the main reason we think is because there's a lot of cell cycle difference between S2 cells in culture and taking an actual organism. And of course, we're looking at different types of damage, irradiation versus single breaks. We could, like for example, take tissues with different cell cycle profiles and see whether there's a different dependency in that case on HR, non-homologous end joining. And we could also look in the animal and see if the dependency on non-homologous end joining might be DNA damage specific. So all future directions. So, and then I want to thank everyone involved. So first of all, Gary, who's a great mentor in the lab. I want to thank Jen, who's been sharing all her reagents and has been very helpful in the process. All the people from the Carpen Lab, and especially my undergrad, Sean, Tim, and Annelot. And then Greg and Ranjit at Yale, and Eva and Bas at the NKI in the Netherlands has helped tremendously with the analysis of the breaks, the sequencing. And then I also want to thank some past lab members, Irene, Aki, and Elaine, to help in the start of this project. And I want to thank you for your attention.
I was really struck by the fact that your homologs are being dragged along. So do you see like chromatin remodeling in trans, or do you see modifications in trans?
No, we haven't looked at it yet, but yes, that's definitely but I mean, I haven't done actually the live analysis of the 2 homologs moving together. But it's very likely that is what's happening. But we haven't looked at specific chromatin modifications yet. Yeah. But it's a future plan.
So let's thank Aniek again. It was great.